Hello guys, today we're gonna talk about excitation contraction coupling. So what is excitation contraction coupling? Basically, it's a process that converts an electrical signal into muscle contraction via Ca2+. First thing first, a nerve impulse reaches the synaptic terminal of motor neuron, and this will cause the ACH to be released into the synaptic cleft, and it will diffuse across this synaptic cleft, bind to the receptor. Once the ACH is binds to the receptor proteins on the sarcolemma, it will trigger an action potential in the muscle cells. Then the action potential is spread along the sarcolemma and move down to T tubule. The action potential reaching the T tubulus will stimulate the sarcoplasmic reticulum, which is SR, and the Ca2+ inside SR will be released to the plasma membrane.
So now we're going to the step 4, which when the Ca2+ is released into the cytoplasm, it will bind to the troponin. This troponin will change its conformation, and this will pull the tropomyosin away from the myosin binding sites on the actin. Normally, the tropomyosin will block these binding sites and prevent the contraction. So once it is moved, the binding sites are exposed, which allowing myosin to attach. So in this process, the key idea is you can think that the Ca2+ is a key which helps to unlock the binding sites. So next, the cross-bridge formation and contraction. In the cross-bridge formation, the myosin head will attach to exposed binding sites on the actin, and this will form a cross-bridge. Then, during power stroke, myosin head bends and pulling actin filament toward the center of the sarcomere. This will shorten the muscle and causes the contraction to be occur. Then, when the cross-bridge detachment, a new ATP molecule will bind to the myosin head, and this causes the myosin head to detach from the actin. Lastly, during reactivation, ATP is hydrolyzed from ADP and phosphate group. Energy release recalls the myosin head to its original position. The cycle will repeat as long as Ca2+ is present and ATP is available. Step 6. When the stimulation stops, Ca2+ must be removed from the cytoplasm. And now, ATP is playing an important role in actively transporting the Ca2+ into the sarcoplasmic reticulum. Last, with less Ca2+ in the cytoplasm, Ca2+ will detach from the troponin, and the troponin will return to its original shape. Tropomyosin moves back to cover the binding sites on actin. Therefore, myosin cannot longer bind. The cross-bridge formation will also stop. The muscle fiber will relax and returns to normal length. So in this process, you need to know that with no Ca2+, the binding sites will be blocked and there will no contraction occur.
So now, let's do a short recap. This arrow is represent action potential. So first, action potential on the plasma membrane of muscle fibers, which is sarcolemma. It will move down to the T tubulus. Once it reaches the T tubulus, it will stimulate the SR, sarcoplasmic reticulum, to release Ca2+. And the Ca2+ is playing an important role in the actin-myosin interaction, which is the cross-bridge cycle. So that's all. Thank you.